Good afternoon, everyone. I'm going to talk uh, about the utility of offering combined PGT-A and PGT-M using a laboratory perspective. So, the seriousness of the situation is why do we need a PGT-A or PGT-M? PGT-A is pre-implantation genetic testing for aneuploidy, and PGT-M is pre-implantation genetic testing for monogenic diseases. So why do we need these tests? So the prenatal screening and diagnosis is very critical, looking at the global scenario of around 6% worldwide births which have serious birth defects, and out of which 50% of birth defects are unknown. And same is the case with, as far as the Indian context is concerned, there is a huge burden of genetic diseases in India. And given the inadequate uh, resources and disease management in India, it is much more prevalent as compared to Western countries. And diseases like Down syndrome, beta thalassemia, muscular dystrophies, which should have been a part of the primary healthcare services, are not there. So the burden is very huge in India. So, in an IVF setup, every second counts for the developing embryo when we are looking at providing a healthy baby to a couple who has come for in vitro fertilization. And if the embryo has any chromosomal abnormalities, the fetus is less likely to implant. There can be a lot of miscarriages, and it may develop into a genetically abnormal fetus or an abnormal baby. So, in order to give a healthy baby to the couples who come to us for in vitro fertilization treatment, we have to make sure that the embryos we are implanting are of good quality. So, in vitro fertilization is basically a stepwise process wherein there are certain steps that you have to follow for couples where the natural conception is not possible. So you go from ovarian stimulation to oocyte retrieval, and then the fusion of egg and sperm, and then finally the embryo transfer. And before you finally transfer the embryo, we can introduce techniques like reimplantation genetic screening and diagnosis as per the requirement of the couple to give them a healthy baby. So the embryo culture is done up to day 5 to day 6, and embryo biopsy is done, which is around 8 to 10 cells. And then these 8 to 10 cells are tested for chromosomal abnormalities or any disorders in the embryo. So embryo biopsy is one of the critical stages as far as an in vitro fertilization is concerned. So it can be done at various stages, whether it is polar body biopsy, it can be cleavage, day 3 biopsy, or it can be blastocyst day 5 biopsy. So blastocyst biopsy has been seen to have a growth advantage towards euploidy. So that is why this is the most prevalent kind of biopsy that is done nowadays. Polar body biopsy is only for maternal driven chromosomal abnormalities because the paternal information is not available in the polar body. So when the oocyte is developing, it will undergo meiosis and it will expel one set of its chromosomes into a polar body, and which is expelled through the cytoplasm. So the oocyte that we have has only the maternal information and will give you information only if there is any inherited disease which is coming from the mother. So this is how a blastomere or a blastocyst biopsy is done. So you have a 6 cell or 5 cell stage, and from that you are taking out few cells, around 8 to 10 cells, and doing this under a laser guided instrument. The blastocyst basically has an inner cell mass, which is comprises of the fetus cells, and it has a outer layer of trophectoderm, which will ultimately form the placenta. So this is the layer that we use for the biopsy, because we do not want to disturb the inner cell mass of the fetus. So that is why a trophectoderm biopsy is done, wherein we remove some 8 or 10 cells from the trophectoderm and send this for laboratory analysis. So in a conventional IVF, you would have an egg and a spermatozoon. They would fuse together and form a blastocyst. And out of these blastocysts, you would select a good-looking blastocyst and then transfer it into the womb. But with next-generation sequencing and PGT-A, what you do is you biopsy the cells, you identify the genetic abnormalities in these cells, and then find out whether the embryo is a euploid or an aneuploid. And then after screening, it is finally implanted into the female body. So, which means that genetic evaluation prior to embryo implantation is very critical, because morphological criteria are very insufficient for embryo selection. So good morphology embryos do not necessarily mean that you will get good quality embryos. So here is where the pre-implantation genetic testing comes in. So you have either screening, where you can do an aneuploidy testing, which is at the chromosomal level. So you are looking at abnormal copy number of chromosomes using the technique PGT-A, or you're looking for any monogenic disorder testing, like any disease that is running in the family, and you want to see whether the mutation is there in the embryos, and you go to the gene level in this technique, and this is called PGT-M. So aneuploidy screening using PGT-A will tell you whether the embryo has aneuploidy or mosaicism, and it will tell you across all chromosomes whether these are 22 autosomes or XY chromosomes. So maternal age is directly proportional to aneuploidy, is directly proportional to IVF failure. So the indications for PGT-A are that women who have advanced maternal age, which is greater than 34 years, there is a previous child with any chromosomal abnormality, there are multiple implantation failures or IVF failures, and there is severe male infertility. And you look for conditions like Down syndrome, monosomy X, which is Turner syndrome, or 13-18 trisomies, Edwards and Patau. If you can see on the right side of the slide, so starting with the diploid number, when the meiosis happens, so the embryo can have either a haploid copy, it can have an extra chromosome, which makes it N+1, or it can have a chromosomal deletion, which makes it N-1. So when it is mating with a normal gamete, so N will become 2N, N+1 will become 2N+1, which is trisomy, and N-1 will become a monosomy. So this is the result of trisomies and monosomies in embryos. On the other hand, pre-implantation genetic diagnosis will go to the gene level. So in PGT-A, we are only looking at chromosomal abnormalities, but in PGT-M, we are going to the specific gene which is known to cause any heritable disease. So it can be sickle cell anemia, cystic fibrosis, Huntington's, beta thalassemia, etc. So the picture on the right side is a typical example of beta thalassemia, wherein the couple is carrying a beta thalassemia trait. So both the parents are carriers of the trait. So there is a 25% chance that this carrier trait will be transmitted to their children. So it can be either a normal baby It can be a carrier baby, or it can be an affected baby. So, in order to understand this, we adopt the technique of beta thalassemia. We adopt the technique of pre-implantation genetic screening for monogenic diseases, so that we give the couple an embryo which is free from beta thalassemia gene uh, mutation trait. So, indications are parents who carry the gene mutation. There is a pedigree or a family history of a genetic disorder. These can be autosomal dominant or autosomal recessive. This can be X-linked disorders which are coming from the mother, or the parents are carriers of a mutation, or HLA matching. So HLA matching is very critical because they would like to select embryos that are histocompatible with any live siblings and might serve as a savior baby for the live sibling. So using both these techniques and a small sample of an embryo biopsy, we can carry out the techniques of both PGT-A and PGT-M. So you just have to remove the embryo cells, you put them in a lysate, you do whole genome amplification, uh, and also a target enrichment based on the site that you are targeting, and to do a low depth NGS and generate the genome sequences. And these genome sequences will tell you whether it is a single nucleotide variation for any monogenic disorder, or it is at the chromosome level, you're looking at the copy number variation for aneuploidy detection, or you're doing the linkage analysis. Linkage analysis is basically gene hunting technique that traces disease causing genetic markers of known chromosomal location that are co-inherited with the trait of interest. So all these can be done using the simple biopsy material that you did from the embryo. So what does this give us? It gives us embryo viability, which is the most critical for an IVF setup. So you have improved implantation rates, you have almost reduced miscarriage rates, a decreased risk of abnormal offsprings. You only look at blastomere and profectoderm samples, chromosomal and segmental aneuploidies, and the technology is validated. So the combined workflow is basically very simple, starting from the same embryo biopsy material. You are doing all the NGS workflow that is required, from library preparation to amplification to enrichment, chip loading, then sequencing and data analysis from simple embryonic biopsy that you did. So there is no need for any other sample, and the whole genome amplification can be done using next generation sequencing, and the same sequences can be taken and the same material can be taken, and both PGT-A and PGT-M can be performed on the same sample. So this essentially makes the life in laboratory very simple, because although the starting material is very low, you are getting so much more information out of this little starting material that you started with. So this is the robust technology that we use, and we're getting both the information at the chromosomal level as well as the genetic level from the same starting material. So how does the data analysis look like? So this is a PGT-A data analysis which has been generated at gene strengths. So you have this embryo which is completely euploid embryo. You will see the embryo's number on the bottom of the on the y-axis, and you have the copy number on the x-axis, and then you look at how the chromosomes are placed and see that all the chromosomes 1 to 22 X and Y are falling at 2 level, which is 2N, or making it a diploid embryo. So this embryo is very good for implantation and will definitely lead into a healthy baby. Then we have cases where, although this euploid embryo is suitable for transfer, there may be some wherein you have a trisomy 21, which is a gain of chromosome 21. And this is an abnormal embryo. And if you implant this embryo, it will lead to Down syndrome. Similarly, you have a chromosome 13 loss here, which is again an abnormal aneuploid embryo with a monosomy 13. So these two embryos are not suitable for implantation. Whereas the normal embryo with a 2n euploid number is completely suitable and can be transferred. So now this is a case study for a monogenic disorder, beta thalassemia gene. The cure for this is only hematopoietic stem cell transplantation. And the parents of this couple were carriers of beta thalassemia gene. And they had a son who was affected already. And the aim of this study was to get a beta thalassemia unaffected embryo. So, and the result that we got was 10 by 10 HLA matched sibling. So what we did basically was a proper protocol that we followed, starting from genetic consultation, taking of history of the couple, then development of a probe against the beta thalassemia gene along with HLA typing, fusing of the sperm and the egg, cultivation on day 5 to make of the embryo, and then doing an embryo biopsy. Same way, taking few cells from the embryo, and then after taking the biopsy material, the embryo is frozen till it is further used. And this biopsy material that we have is sent to the lab, and we perform PGT-M and PGT-A, both procedures, and finally select the best one for the couple and do the embryo transfer. Within a span of 7 to 8 days, the entire laboratory procedure is complete, from running of the sample to data analysis. And this is a very fast technique which does not take more than 2 to 3 days for doing the laboratory work and around a week for analysis. So the total blastocysts that we got were 18, out of which 2 were normal, 8 carrier, and 10 were affected. When we looked at the normal embryos, they did not match with the already available sibling. So we were left with no choice but to go for the carrier embryos that were there, and out of which 2 matched, and 1 was a complete HLA typing match with the already present child. And this could be used as a savior baby by stem cell transplantation, and could save the baby that is already affected with the disease. So this was another case with the same HBB HLA typing was required. But here, as you can see, that we have the female as well as the male, which are carriers, and we have an affected child. But when we used around 4 embryos, you could see that most of the spaces here are vacant. This is because the embryo quality was not good here. So that is why we have made this SOP in our lab, that whenever we are looking for embryos for PGT-M, what we will do first is, after doing the embryo biopsy, we will take them through PGT-A first, look for the euploid embryos which are free from any mosaicism or any deletion duplication, and then use these embryos for PGT-M. By doing this, the success rate of the technique has been increased to a lot more higher percentage. So to summarize, the 2 techniques, PGT-A and PGT-M. So these are for PGT-A is for abnormal chromosomal copy number detection. The technology can be used are FISH, RACGH, or NGS. And the indications are advanced maternal age, recurrent pregnancy losses, repeated IVF failures, or infertility. Similarly, for the monogenic disorders, it can be single gene disorder detection. The techniques used are specific probe primer, PCR, Sanger sequencing, STR markers, and the indications are known: familial monogenic disorders or HLA typing. So combining these two technologies, PGT-A and PGT-M, using NGS, will give you euploid, unaffected embryos and ensuring a higher success rate of IVF implantation and disease-free, chromosomal abnormality-free, healthy baby. So the take-home message is that IVF using NGS-based PGT and PGT-M has completely transformed the area of reproductive genomics, and the integrated use of these techniques is efficient, economic, safe, accessible, and reduces the rate of passing the genetic disorder to fetus and promotes approximately greater than 70% chances, which is the percentage that we get in our center, and chances of a healthy pregnancy and take home baby. With this, I conclude with our recent research. These are some of the papers that we have published in some journals. So we're looking at mosaicism primarily, and we saw that a mosaic embryo can be completely transformed into a healthy baby using the self-correction technique of self-correction hypothesis of mosaicism. And whatever implantation and of embryos we are doing in the IVF setup, it's based on the PGDIS guidelines. So PGDIS has published some guidelines which we adhere to the book, and resulting in a very high percentage and high success rate of our IVF cases. So thank you. I'm open for questions.